Here is the next lecture on protein composition and structure. And so as a reminder from the previous lecture, the hierarchy of protein structure is 4 different levels. The first level is the primary structure, which is the sequence of the amino acids. And then we have a building block to get to the secondary structure, which is small units of that repetitive sequence of amino acids. And then that is the building block for the tertiary structure, which give it its overall 3D shape. And then from there, you build up to have a building block to a quaternary structure, which is more than 2 polypeptides. And so we're going to just dive deep into each of these structures, starting off with the primary structure, which is peptide bonds and disulfide bonds, as I mentioned previously. And so peptide bonds are planar and have partial double bond character. And so right here, that is demonstrating that here with looking at the resonance structure. So you have the peptide bond that's right here because we had that carboxyl group attached to the amino group, but that double bond with the carbon and the oxygen can delocalize to be between the carbon and the nitrogen. And so that's what we talk about, this having this partial double bond character. And then also almost all peptide bonds have a trans configuration. And so if you once again look at this peptide bond that is highlighted here, because this is a carbon bonded to an oxygen, and then you have the nitrogen and the hydrogen. So that is your peptide bond. And whenever this trans configuration, we don't have any sort of steric hindrances that's happening, but if it ever gets into a cis type of configuration, then there is some steric hindrance that is happening. But of course, this is not always the case. Like always, there are exceptions, and so the most common cis peptide bonds are found between proline and other amino acids. The steric differences between the trans and the cis forms are minimum in this sort of situation, and both can form. And then also there is some rotational flexibility that can happen where the alpha carbon right here, which remember the alpha carbon is directly connected to that carboxyl group that was there. So this is the alpha carbon, and then from there you can have rotation that is happening on the bonds connected to that alpha carbon. And then now going into the secondary structure, and for the secondary structure, recall that you have 2 options. You have the beta sheets and the alpha helix. And so this is the demonstration of the alpha helix, and you can actually with alpha helix, you can have the right hand helix or the left hand helix. You can usually kind of tell by utilizing your thumb and the direction that your thumb is pointing towards. It gives this its characteristic of being labeled as a right hand versus being labeled as a left hand. And remember that this quarreling interactions is due to the interaction of the carboxylate group with the amino group. And so these are all hydrogen bonds. The direction of the rotation about the axis of the alpha helix is also called a skew sense. And essentially all alpha helixes found in proteins are right-handed, which is less steric. And then, as I mentioned, we also have these beta sheets, which is planar. And so the arrows here is pointing towards the carboxy terminal end. And this end is the amino end, so where the nitrogen is located. So nitrogen, carbon, and that's the direction that it's going. You can have anti-parallel beta sheets and you can have parallel beta sheets. And how you can tell is, once again, remember that beta structure with nitrogen to the carboxy group. You can see right here you have the nitrogen group, and then as you go to the left, then it runs into the carboxyl group, which is the carbon bonded to the oxygen. And once again, nitrogen to the oxygen. So it's going in this direction, going left. But for this one, if we find the nitrogen first, and then from there we see the carbon bonded to the oxygen, that's the C terminus end. And so we're going into the right direction. One way to figure out whether or not you're going in the wrong direction is if that nitrogen, if as soon as you go to the left direction, is directly bonded to the carbon with the oxygen group, so the carboxyl group. That is the peptide bond. So we don't want to go in that direction. We want to go to the actual unit of the amino acids. And so that's the N terminus and going to the C terminus. N terminus going to the C terminus. And so since these are opposite directions, that makes this anti-parallel. But when you have them going in the same direction, this is the parallel beta sheets. So once again, find your nitrogen. If you're going to the right, you see that this is not the carbon that's bonded to a oxygen, so we know this is not the peptide bond. So we can continue on, and we see here we finally have the carbon double bonded to the oxygen, and so we know this is N to C, so it's going in the right direction. This one right here, nitrogen, there's the carbon right here, but the double bonded to the oxygen, so it's going to N to C, so it's going in the right direction. So they are both going in the right direction, making these parallel beta sheets. And of course, they got to make things even more complicated, where you can have a mix of beta sheets, where you can have parallel mixed with an anti-parallel, and have a combination of each. And so with these protein structures being secondary, you can see it is the N terminus to the C. And so you always have this head to tail type of connection. You can't have the head to the head connection. And so in order to have this head to head connection of this sort, you have to have in between a alpha helix, and then from there now you can have two of the beta sheets that are connected. And so having different pieces of a secondary structure, this mixing is called motifs, or called super secondary structures. And some common names of these motifs are found right here. Going into seeing them a little bit better as far as examples, you have beta, alpha, beta, which we just had example of earlier. We have a beta sheet and you have an alpha helix that's connected to another beta sheet. Then you can have the anti-parallel, where you have that head to tail action going on. That's a beta hairpin. And then you can have two alpha helixes that are connected together. Of course, they can have different sequences of amino acids, which makes them different and thus having that motifs or a super secondary structure. And then you can also have this beta alpha, which is this Roseman fold. And then you can have two alpha helixes with different sizes. And those are pretty much the main super secondary structures that we will cover. And then within these motifs, you can have this alpha beta barrel, which is once again example of a beta alpha beta loop that actually creates this barrel type of shape, and that's why it has this alpha beta barrel type of naming nomenclature. Then going into tertiary structure, this right here is a schematic drawing of a myoglobin where you have a hemi group, and in the center you have an iron atom that carries oxygen, but we will talk about that more later on. But this, as far as main techniques to kind of characterize and reveal the details, is typically the X-ray crystallography and NMR. And then there's all sorts of representation of this 3D tetrary shape of the protein. You can have the ribbon, you have surface contour, and then ribbon with side chains and space filling. And these are the schematic representation of a 3D model of a tetrary structure of the protein. And then these tetrary proteins can have different classification, where you have all alpha helixes, and that's alpha. Then you have all beta sheets, that's beta. And then you have a mix of the two with alpha and beta. And then you can have this barrel type of formation, where for this example, these are all beta sheets that forms this barrel type of shape. And then last but not least, we have the quaternary structure, which is makeup of these tetrary structures that is interacting with each other by either hydrophobic effect, hydrogen bonds, salt bridges, or van der Waals, which is in other words called dispersion forces. And so to summarize everything as far as the protein structure, we first have the primary structure, which has a specific sequence of amino acids. And then from there, they can start to interact with each other with hydrogen bond to form the secondary structure, which can be an alpha helix, which is notated here, or a beta sheet. And then those can interact to finally form these polypeptide chain, which is also the 3D shape of the protein to have this tetrary structure. And then these tetrary structure are units for making up the final level, which is the quaternary structure, which is the final assembly that we will discuss within this course. And that concludes this piece of the lecture.